2, 1, go.
Hi, my name is AMorris, and my project is on determining the binding capabilities of protein kinase C delta, or protein kinase C inhibitors in protein kinase C delta. Just a little bit of background. Protein kinases are essential for substrate activation, which means that once they are activated, they'll go on to activate other things such as transcription factors. There are three conserved domains in PKC. There's the regulatory domain, which is over here. There's the catalytic or kinase domain in blue, and the hinge region is right here. PKC delta is a serine-threonine kinase. It's a novel isoform of PKC, which means that there are some changes within the amino acid sequence. PKC delta 1 and 8 are the human variants. Alternative splicing will determine the function, whether it's apoptotic or pro-survival. PKC delta 1 is apoptotic and PKC delta 8 is pro-survival. What makes PKC delta so special is that there's a lot of novelty within the C2 domain, and that means that it's calcium independent, whereas most protein kinase Cs are calcium dependent. This is because there's a change in the amino acid of aspartic acid to a histidine residue. This is just another picture, a more detailed picture of the domains. A little bit more background. Activation will follow the PIP2 pathway, or the phosphoinositol pathway. So it's going to be DAG and calcium, but in PKC delta's case, there will not be calcium needed for activation, or PMA, which is formyl maestrate 13 acetate. Cleavage at the hinge region is necessary for apoptosis to occur, and that is where the arrow is. My methods. There are no known inhibitors of PKC delta. So what I want to do is determine inhibitors from other PKC isoforms. I'm specifically looking at these two, GF109203X and G0983. These inhibitors bind to the ATP binding region, which prevent activation. I want to compare the structures of the different isoforms of protein kinases because there is no known inhibitor of PKC delta. I'm going to need inhibitors from other PKC isoforms, and comparing the structures will allow me to determine whether or not binding can occur, because I'm going to be comparing the amino acid sequence. And I'm going to do this with the use of a protein model using the programs JMOL and SwissProt, and this is going to allow me to overlay each isoform, and that will create a homology model which will allow me to determine whether or not the sequences are similar enough for binding to occur. And I'm hoping in the spring to test the effectiveness of the inhibitor that I choose within the lab. My materials are going to be JMOL, which is a 3D protein modeling software, SwissProt, which is a free online resource, and that's going to create the actual homology models. It's going to overlay each JMOL file that I make, and Uniprot, which is another online tool that's going to align the sequences of the amino acids and the protein. So because all of my tools are online, the cost is $0. And for the conclusion, what I'm planning on doing is determine if inhibitors of PKC isoforms will bind to PKC delta. I want to mutate some amino acids within PKC delta to see if that prevents binding of the inhibitor. I want to determine if the apoptotic function is abolished once the inhibitor is attached. And I hope to eventually model the catalytic domain based off the homology model. There are some implications that make research in inhibitors with PKC delta very important. That includes neuronal survival in Alzheimer's, a decrease in tumor proliferation, and prolonged cell survival. Dr. Shore, Dr. Ellis, Dr. Patel, Dr. Gonza, the people in Milwaukee, Dr. Fonzen, and our dean, Dr. Simoto.